The Scinus Bioreactor is a closed system for the controlled cultivation of cells, supporting research and development, as well as good manufacturing processes. The system operates with a touchscreen interface and is compatible with industry-standard controller software for process control, monitoring, and data acquisition. The temperature-controlled hardware houses the single-use bags on a unique rocking platform that provides an optimally homogenized culture environment with minimal shear stresses. It enables true scaling in culture volume thanks to a unique roller clamp system expanding to relevant amounts in one culture run. Combined with either the single-use adherent or suspension bag, it provides a scalable system for large fold expansion. The adherent culture bag is used for the cultivation of adherent cell types on microcarriers. The bag can be expanded up to a surface area of at least 35,000 square centimeters, which equals 200 T175 flasks. 2 filter pouches of 100 micron prevent the microcarriers from entering the perfusion loop. The suspension culture bag is used to provide a volume-scalable culture environment for the cultivation of suspension cell types, such as T cells. Here, 1 micron filter divides the bag into 2 compartments, where the lower is hosting the cells and the upper is used for medium perfusion. Both bags have integrated sensor technology that measures oxygen, pH, and pressure at the site of culture. The Carboxygenator is a patented oxygenation system that, through a continuous feedback loop with the culture bag, ensures that the predefined culture environment is maintained. The addition of gases takes place through a gas-permeable tubing. With our Carboxygenator, we can provide your cells with the oxygen they need without the use of a headspace, minimizing any additional shear on your cells, as there is no air-liquid interface during the rocking process. The versatile tubing set with weldable tubing makes it easy to add medium or remove waste. By adding an extra bottle of medium to the perfusion loop, the total effective volume of the bioreactor can be increased. At any point during the culture, a representative sample of your cells can be taken, all while maintaining a closed workflow and without interrupting the run. After analyzing the sample, the Scinus can be adjusted accordingly if desired. The interoperability with other upstream and downstream technologies allows users to easily integrate it into their existing processes and systems. Multiple adherent and suspension applications are feasible. Scinus Cell Expansion provides an innovative, controlled, and cost-effective solution to automate your cell culture processes.